here we are. And I sort of lost my voice yesterday, and it's not because I'm sick, it's because I'm taking care of a cat, and I have one of my daughter's cats, and I have an allergy to cats, and I've been hanging out with it for 2 weeks, and I'm like, why is this a good idea? Anyway, so there, that's going out in the World Wide Web, and I hope she sees that in Hawaii, where she's on a belated honeymoon with Jeremy, and I have their cat. And their cat's name is Katahdin. Okay, here we go. Onward with our course. I got distracted by a class, one of your classmates. So, following up on the first exam, I'm still talking to students about that. That's good. Then, Riley's not here. I guess he's still sick. I didn't even hear from him this time. He's really good at notifying me, but I didn't even hear from him. So Riley's actually sick. Then we're rolling in chapter 18. We'll probably finish that up today and start talking about 19. And 19 is a quicker chapter for us. There's a lot of really important stuff, but it flows through, and then we go on to 20. And 20 has lots of stuff in it, but it's tying it all together. It's officially the last chapter in our textbook. When we're done with it, we'll go back to earlier chapters that we skipped over. This should say those of you who need to write a term paper need to start working on it, and it's described in our syllabus. So on Tuesday, well, I'll put that in the link today. I'll put a description, but the details, the rubric of the term paper is described in there. You're going to pick a receptor and explain certain features of it. Yes, Catherine?
Just a quick question about that.
Yep.
Does it have to be like a receptor that we hadn't really talked about, or can it be one that we've discussed in class?
Yep, you could pick the estrogen receptor. We talked about it a bunch. Yeah, when I'm reading it, I'm gonna know that, and so I would just expect if we wrote one about an estrogen receptor, there would be good information in there, because we knew a lot about it, more like that, So I just make that adjustment. Yeah, but you're welcome to pick any receptor. The students that are doing the 4-H STEM Ambassador Program, they do nothing. They are just doing that, and their purpose or their requirement is to complete the program with Sarah Stark's approval. And if you do it, she's like, yep, they did fine, right? So that's what they're up to. And so when I talk about the term paper, those students don't have to do it. It was an option at the start of our course. And I think it's a very good idea when you pick a receptor, write your term paper about, is just to send me an email or ask me before or after class and say, what about this receptor? And I'll be like, oh yeah, and I'll probably have a couple of comments, a couple of suggestions about something, that would be a nice thing to kind of focus in on like that. But again, everything is in our syllabus like that, describing it, and I'm happy to talk to you repeatedly about it. It's a science term paper, so it doesn't have a specific format, but it's a style of science where it's certainly no plagiarizing. And just as a side note for no plagiarizing, it's so easy to write now that you can do just voice to text stuff. That's the way to write. When a person has writer block, they're sitting there with a blank piece of paper and they're not able to write it down. So you can always say it, and even if it's lame, you can just say how you understand it and turn that into text and then clean that up without stealing, And science is written so carefully that it's just, students are just desperate to just copy and paste like that, just copy and paste. And, there's no way for me to know if you use some kind of chat GPT thing. I guess I don't know anything about that, but hey, don't, because it defeats the purpose of your education, like that, where you're like, this AI is getting smart, and I'm paying for it, right? So definitely we could talk about it, and should be a satisfying assignment rolling with the kind of writing and learning we're doing, like do, like that. Okay, so that's where we're at. Anybody have any other questions about that or anything at all? Yes?
In the textbook, chapter 18, there is a phrase that's interesting, or like sentence that's just confusing to me.
Are you looking at the page right now?
I think.
What page is it?
It's 604. It says a typical.
What is it?
604. Beginning of 18.
Yeah.
A typical eukaryotic cell cycle is illustrated by human cells and culture, which divide approximately every 24 hours. And I know in class we've been talking about how brain cells are differentiated, so they're not going to be going through this cell cycle. So I'm just confused why they're saying, like a typical eukaryotic cell is doing it every 24 hours. And is it just because it's like in a culture that that's the rate that it's happening, or like?
Yeah, what's going on there is, you know, a lot of times the book will, you know, they'll insert something like that and they don't, you know, explain it more fully. And they're just throwing that out there as a kind of an average thing. And we talked a little bit about how non-mammalian embryos divide really fast. They don't even have a G2 or M like that. And so the cell cycle varies. And what does that mean? So start is the G1 to S transition. That's where this red light is. And it's red because the CDKs are inactive. And to turn the light green, we need maybe cyclin is the rate limiting step. Gotta synthesize some cyclin, activate the gene. Or maybe there's some small inhibitory proteins we're gonna learn more about today. Or maybe there's some phosphorylation that needs to be altered to turn the light green. So it's literally biochemistry, you know, that needs to happen, right? Like that, okay? So that's the start of the cell cycle, and the length of the cell cycle, it simply means it goes around and stops again. So that's the length of the cell cycle. And they're saying 24 hours in humans. It varies a lot. It varies in us when we're embryos. It doesn't get really fast like a fruit fly or like a chicken egg like that, but it gets faster or slower, and some of our cells divide faster or slower. And you can establish that for every part of your body right now. You know, what's a cell cycle in our healthy skin cell like that? How long does it take, right? What about under a scab? How long does the cell cycle take under a scab, right? And there are two things that people, you know, think about. One is just how long does it take to get around the circle like that? How long does that take? And the other is really the thing I was talking to some student about. When we say, a lot of times when we say, oh, the cell cycle is going fast, it doesn't actually mean that it's going around fast. It means it gets to go frequently. So like when somebody says, oh, this tumor is growing fast, doesn't really mean that the cells are advancing through the cycle fast, but they just get to go through it frequently. So that's a little clarification like that. And that's what's happening in the skin. The skin cells are growing slower, and then under the scab faster, just because of the frequency they get to go through the cell cycle. So like that. And then you could start breaking it down. You say, oh, it takes about 24 hours. And people say that because it takes a sort of specific length of time to get through mitosis for a human cell. And it's like a couple of hours that it takes, like that. And then S phase with our genome takes a while, and it's about 6 hours, like that, you know? So there's some kind of limits to how quickly it can go through. And so there's a lot kind of loaded up in this, and it's about 24 hours.
Okay, so they're kind of saying, like, if a cell is going through the cycle, then it might take 24 hours?
Yeah.
Okay.
Yeah, if it goes around, it might take, kind of 24 hours. And that's a kind of classic number for humans, is like that. And then for some other animal that's dividing fast, like in lab class, we looked at the caterpillars that's growing into a moth, and those cell cycles are fast, right? And they're both getting to go through them frequently, but they're also pushing that, going through it quickly like that. And those might be 10 hours like that, you know, so it varies like that. Yep. And that number is not important for us, but just kind of the concept that we're just talking about, just it satisfying or whatever to understand, especially the principle that the rate of the cell cycle is really typically how frequently they go through start. Yep.
When we're talking about like the cell signal transduction pathways, like if we talk about the RAS pathway, is that happening in G2 because they're producing ALK, which produce cyclin?
Yeah, that's a good question to talk about. And people would say that in mammals, and more broadly vertebrates, G2 is just a sort of generic time that the cells really typically don't spend a lot of time there. They got to go through start, so they replicated their DNA, so they finished that, and they're like, I could divide, and they kind of just hang out there in G2 kind of for sort of a generic few hours, like that. And so there's gene expression happening, their cell is living, but it's a kind of a vague time for mammals. In some, and other vertebrates, in some invertebrates, they actually spend a lot of time in G2, and they actually, really can pause the cell cycle for a really long time in G2, like that, like insects and other invertebrates like that. That's more prominent. But, and so we spend, vertebrates like us spend, we think of the cell's life as being G1. It's G1, and that includes the G0, the red light. That's when the cell's doing all that, and so that's where the cells are when RAS gets activated. They're in G1, and a lot of times they're already at the red light. They're always just waiting for the rate of thing like that. Other times, some growth factors activate RAS earlier in G1 because there's some different things that people are learning more about G1. There's kind of some interesting features of G1, but we think of cells, animal cells, and certainly vertebrate cells that's living their lives in that G1 period, like that. Yeah. Other questions people have about anything at all? Yeah, and I apologize about my voice, but it's just been getting more and more raspy, and this cat really wants me to pet it. And I'm just like, buddy. Yeah, so we were doing a lot of that this morning. I got coffee and stuff like that. Okay, so we showed this before, and we're talking through these three pieces. We talked about the regulation of start, and there we really were emphasizing, we're going to have to make more cyclin. And cyclin's a protein, and so we're going to have to activate that gene. So we got some transcription factors that we love that are going to activate that gene, and they're going to be regulated by signaling pathways and signal transduction like RAS. So now we started talking about this, and this is just a brief package, and it's at this other point in the cell cycle. And it's good sometime to appreciate, and I talked about this briefly before, but here's the cell cycle always drawn like this, and we love start, and we're over here, and we're talking about the G2 to M transition, which is a DNA damage checkpoint. And when people say, what DNA damage? There actually can be still some issues left over from S phase that still haven't been resolved. So it's kind of like, in some sense, G2 is just kind of like getting stuff kind of cleaned up. It can last for a while, but the G2 to M transition is a DNA damage checkpoint. The cell is going to look internally like the washing machine looking by itself to say, the lid is up, I'm going to stop until the lid gets down. Okay, so we're looking at that, and you can see people would say, no, we're way over here. Like, right, we're not talking about start right now, we're way over here. And it's really kind of a misnomer to say we're way over here, because what we should do is we should draw this cell cycle out like it's a line of time. And so instead of saying we're way over here, we should say, we're at a very different point in time. Over here, yes, CDK and cyclins are regulating entry into mitosis. It's a different members of the families of those genes. So we're at a different time in that cell. And the story we're talking about is really neat and straightforward, and we're just going to finish it. I just have some review stuff. I'm going to skip over that since we already kind of dove into this. And so for this regulation of entry into mitosis, people like to go to that time in the cell cycle because it's really a simple package. We don't have a story for the small inhibitory proteins. We have a story for a cyclin, but it's already made. Cyclin builds up in G2. This thing is ready to go. The issue is going to be that it goes. In a healthy cell, everything's good. It just rolls forward, and it's on its way. In a healthy cell, there are these two inhibitory phosphates, and those need to get removed. And we learned last time that the guys that remove phosphates are called phosphatases, and this one is called CDC25 phosphatase. And every eukaryotic cell on our planet has an ortholog of that, and it's its function. Its function is just remove those inhibitory phosphates so we can go into M phase. And this will stay active throughout M phase, okay? So, when there's DNA damage, what happens is CDC25 gets inhibited. So its activity is blocked, and therefore those are not removed. And so if we're looking at a cell that is at the G2 to M transition, essentially where the washing machine lid is up, what it looks like is this here. It's got the CDK, that's the action. It's got the cyclin it needs, cyclin-dependent kinase, but its activity is blocked by these two inhibitory phosphates that need to be removed, but CDC25 is inactive. And it's inactive because it gets phosphorylated. So when CDC25 is phosphorylated, it is inactive. And there's a simple figure in your book that shows this pathway. And so I'll just show you that figure right now. And it's just important, it's satisfying the book did this because the book is showing two things at once. And here's our pathway that we're talking about right now. So there's DNA damage, and CDC25 gets inactivated, and so CDK1 stays inactive. So that's the DNA damage checkpoint at the G2 to M phase transition. And it's just showing that. What's new to us on this figure is there's a kinase that phosphorylates CDC25, and the kinase is called CHEK1. There's this other pathway, and it's a totally different story that we'll see later today. So we just really want to circle that because there's a nice little thing of dominoes falling, right? The DNA gets damaged. How? Well, look at that one. Unreplicated DNA. We just talked about that. If S phase is not done, you're like, the cell is like, hey, loser, don't divide yet, right? We got to finish S phase. Come on. We can do better than that, right? You got to replicate the DNA and get them all duplicated, right? You got to get that tetraploid G2 set up so they can separate, and you're going to get two good cells. And it would be a really huge mistake to divide cells when you haven't finished replicating the DNA. So we can remember that especially. The book likes to emphasize that there are certain types of DNA damage that are likely to make this thing active. But it gets activated, detecting it. We don't need to know the details of that, but just proteins are detecting that damage or unreplicated DNA, and that activates CHEK1. And CHEK1 is a kinase, and it phosphorylates CDC25 and inactivates it. There would be orthologs of these proteins in every eukaryotic cell on our planet. This is just a little switch, and you're just like, oh, I see, I understand that. If you haven't finished replicating your DNA, don't divide. Right? It is a thing where you're just like, come on, you can do better than that, right? Like that. Yeah. And so it's inactive, and it stays inactive until the damage is replication resolved, and then all those get reversed, and CDC25 gets dephosphorylated and active, and it just removes those inhibitory phosphates, and that's it. And that's the whole story. That's the whole package right there. Okay? So it's nice. We came to this point, way over here, this time in the cell cycle, because it's just a nice little package, right? It's like the little electric button switch in the washing machine that detects the lid is up. If the lid is up, just stop, 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 stop. Oh, the lid is down. So we just got to know that as our good example. And it's got some phosphorylations on there, and that's where we'll really focus on the role of phosphorylation in regulating the ADK. Yep.
Does it matter if, like, does ATM activate CHEK2, which only activates CDK1, or just the way the diagram is?
So are you thinking about this other part of the diagram? Or just
so
this
on the
part?
bottom, right? So CDC25 is going to turn off CDK1 or CDK2, but the way the diagram reads to me is it turns off CDK2 if ATR is activated, it turns off ATM if CDK1
see,
is activated.
and so this is a kind of a good thing to see sometimes where the person who created this is an artist who maybe has like a degree in biology, maybe, you know, they're an artist, they're a professional artist. And so the author explains it to them, and the author told them for this figure, they said, I just want to smash these two together. And the artist definitely said, do you think that's a good idea? Do you think that's going to facilitate learning? Or do you think somebody like me, Jeremy, is going to be like, I don't really get this? The author should have made two separate figures and just been like, this is what's happening. What I circled is what we're learning right now. And again, we're going to look later in the class, and we're going to circle, it's this other pathway. So it is confusing, because it does look like, well, you could draw a line to CDK2, but this, it's really what's going on here is it's a different time, right? So you can't draw it because it's not there, it's not playing a role. Yeah, so just like recreate this figure, because there's our figure. That's the story for our G2 to M transition. DNA damage checkpoint, and it's sweet, just boom. Like those dominoes fall for those reasons. Then Hannah and then Brooklyn have questions.
So, like, you know how there's like, right before CDC 25, there's like like it indicates that it's getting inhibited with the like, horizontal line, but then right after it has that arrow pointing towards CDK, does that mean it's like you know what I'm talking about?
Yeah, so that's a good question, and just to point out, in these kind of figures, there'll be an arrowhead, and the arrowhead means an activation pathway. So the arrowhead here is CDC25 will activate CDK1 by removing the inhibitory phosphates. Then up here, this block means inhibit. So in this case, check 1 will inhibit CDC25 by adding a phosphate.
Okay, so you're saying that
Yep.
by DHK1 inhibiting CDC25, it activates CDK1.
Nope. Check 1 inhibits CDC25 and prevents CDC25 from activating.
Yeah, see, that's what I was confused, because
Yeah.
it's showing inhibiting, but then it shows the arrow.
Right.
So that's
Yes. Yeah. And you know, I mean, we could think of a real world scenario where you're like, you know, CDC25 is a person who's going to kick a dog, and you're like, don't kick the dog. So, right, it's just a series of cause and effect relationship. And so you could say check 1 inhibits the activation of CDK1 by inhibiting its activator, right? So the kid's going to kick the dog. I say don't kick the dog, but you could put your hand over my mouth. So it is a little bit confusing, but it's also understandable. You just have to take a moment to talk your way through it. Yep. Because of that. That's good to point out, though, these ideas that that's like an inhibitory symbol and that's an activating symbol. Yep. Let's see. Brooklyn had a question.
So CDC25, it inhibits or it activates CDK1 down the line, or is that just like?
Yes, this is like a timeline pathway and a cause and effect relationship pathway, upstream and downstream. We talk about that, steps like that. And so you would say CDC25 is upstream of CDK1 activity. And it needs to remove the inhibitory phosphates to allow CDK1 to do its job. So that's how it activates CDK1. Is CDC25 removes these inhibitory phosphates, and now it's active. So, to do that, CDC25 needs to be active. If there's DNA damage, we want to stop the cell cycle, and we can do that by inhibiting CDC25's removal of the inhibitory phosphates.
I'm just having a hard time visualizing the whole pathway.
Yep. I think a good way to visualize it would be to draw it twice. And draw it once when there's no DNA damage, and show that in that case, this check 1 is inactive. So CDC25 removes the inhibitory phosphate. And then draw it a second time where there's DNA damage. So in this case, you would have a separate drawing like that, and there would be check 1 phosphorylating CDC 25 and inhibiting and failing to remove the inhibitory phosphates.
Yeah, I think it's
Because they're
important.
two states.
I think it's important to note that if CDC 25 is phosphorylated, it's not going to remove the
Yeah,
inhibitory
this
proteins
shows
from
the
CDK.
inactive CDC 25. When CDC 25 is phosphorylated, it's inactive. And that's a thing where you need, on a case-by-case basis, we always have to learn when protein is phosphorylated, is that active or inactive? And in some cases, when a protein is phosphorylated, like elk, it's active. In other cases, when it's phosphorylated, it's inactive, like FOXO. Speaking of FOXO, we can do this too. So we saw from the start of our course, there's this transcription factor called FOXO, and where'd that thing go? And so here's that old FOXO, and here's the nucleus. And when the cell's growing, AKT phosphorylates FOXO, and FOXO gets a couple of phosphorylations. And when that happens, a protein called 1433 binds to FOXO and prevents it from going in the nucleus. So it can't go in the nucleus. And FOXO is a transcription factor where its function is in the nucleus. And if it can't get in the nucleus, it can't do its job.
Wait, what is the purple arrow?
The big purple arrow is just showing either FOXO could go into the nucleus or it can't.
Okay.
Is sort of depicting that. And it can't when it's phosphorylated, and it can't because 1433 binds to it, and 1433 says, you're just standing in the cytoplasm. You're not allowed in the nucleus. And that's where the action is. So look at this reality. Here's where CDK1 is, and CDC25, phosphatase, needs to get in there and move those inhibitory phosphates. When CHEK1 adds a phosphate to CDC25, this protein binds it and prevents it from going into the nucleus and doing its job. And the protein that binds to it is this 1433 protein. So that's just a detail from that figure. And, you know, like, we're appreciating that, although this is our figure, we just want to focus on part of it and draw it, and we want to draw it twice, once when there is DNA damage and once when there's no DNA damage, and mark out which is what's going on with each of those proteins, okay? And then that's the thing. And it's like a different time, the G to an AM transitions, different time, then start. We go to that, and our book goes to that, because there it is. It's like that. And it's the framework, and like I just said, oh, there's more knowledge that can be added. Scientists know a huge amount of this, because remember, this pathway is present in every one of our cells, and every animal cell, and every plant cell, and every eukaryotic cell on our planet, right? It is a profoundly significant pathway for the history of life on our planet. And so, and it also is describing this molecular switching that molecular biologists love to focus in on and see how is that switched going, like that. Yes, okay? So the book is doing that, right? And pretty soon we'll look and we'll draw that other one, but it's going to be a different time because it's going to be a different time in the cell cycle. Okay? Yep. So that's really important, right? We have 3 parts of chapter 18 we want to get to. We love START, talking about START. We want to go over here, regulation of the entry into M phase, to get this, just to get a what is a relatively simple view of this, okay? All right, because now that's our little package, and it's good knowledge to help us, because now we're going to go back to START and talk some more about some more regulation stories in START, okay? And any questions people have about stuff right now?
I
Okay.
think I just, oh, I do just have a quick question. I think I just wrote it wrong, but it's CDC 25, not CDK 25.
Yes, CDC. Yes, yes.
The confusion, I think.
Yeah. Yes, and I can just mention a little bit about this. The scientist that studied the yeast that caused sprinterize was in Seattle, who discovered these first. And he was a geneticist, and he was doing those mutations, and he was doing those temperature-sensitive mutations to understand what the gene products the yeast need to grow. And so he named the genes. And he just named them cell division cycle mutant and a number. And he found a bunch. And this is what he found. And that name is retained. Other names have sort of taken over. I won't just, you don't have to write it down, but he originally called CDK1, he called it CDC2. All of them were just CDC, cell division cycle mutants, and a number. And they were different genes, and he just was studying all of those in Seattle, in his lab, at the Fred Hutchinson Cancer Research Institute. You know, and they're just like, Lee, why are you studying yeast? And he's like, I don't know, there's really pretty striking stuff going on. Like that, yep. Okay, so that's, you know, the origin of that name, a little bit thing. Okay, so now we're going to this other place on the cell cycle, really another time, back to start, back to the future, start, right? So we like to talk about cyclins, right? And the role for cyclins, right, like this, because this is where we see the three things that we want to learn about. So we learn some about cyclins, gotta have them, cyclin-dependent kinase, right? And then there are these small inhibitory proteins, and we want to learn some about them, so we're going to do that now, some. And then we're not going to talk about phosphorylation of the CDKs, because it's got a lot of stuff going on. And we have that nice story. So if we want to talk about phosphorylation of CDKs, let's just go to M phase and say, look, this is a pretty good story, right? So, you know, if you went and took advanced courses and did research on it, you might be doing research on those, because it's a big thing. But it's just complicated because there's a lot of stories of inhibitory phosphates and activating phosphates like that. So we're just not going to worry too much about it, except for that it's part of the story. So now we're going to talk back on this, okay? And we're going to talk about a couple of things. Here's the first one. The book shows this. You don't have to see this in detail, but what's going on here is it's showing some DNA in a cell, and we're at start, so that G1 to S transition, and the S is DNA replication. So this is a timeline, and it shows the DNA starting to get replicated in this figure, okay? And see the yellow? The yellow guys show up early. They're called MCM proteins, and they help the DNA polymerase show where to start and get going. So see them showing up early? Like that. And look at who helps get their job done. So this is the other thing to see on this figure. So it's not happening. We're not in S phase. We're in G1. And now we're going to go into S phase. And here's who helps. There's a CDK cyclin that's phosphorylating the MCMs, helping to activate their function. So literally, well, let me just go to the other point in time. Remember, I said at the G2 to M transition, when the CDK activates, we learned it phosphorylates histones and causes the chromosomes to condense and become visible. It phosphorylates nuclear lamin proteins and causes them to disassemble, and the nucleus temporarily breaks down. So there's just this direct biochemical thing. These CDKs are active. Here's what's happening. They're active. We're going to start replicating DNA. You know, and it's satisfying to see that. There's no additional magic, and you will learn about that if you go to grad school. It's this. It's just boom. We're just replicating DNA. Let's go. So I just want to show you that, because that's what the purpose of that figure is to show you that. And just to show you this, because we can learn the name of these MCM proteins, and just see MCM are involved in the initiation of eukaryotic genome replication. MCM, like that. And just that image, that image of us seeing the CDK cyclin phosphorylating this, right? The light turns green at start because CDK gets active. And here's an example. It phosphorylates MCM and the DNA starts replicating. So it is really the G1 to S transition. It's like, let's go. So I just want to show you that figure. Then, next, we're going to go through this list and just focus on these stories from this list for our course. So we talked a lot about transcription activation of target genes like cyclins and the guys that activate them, right? So we did that one first, and now we're going to talk some more. And these are just little short stories, but each one tells another piece of the start. And the first two of these, retinoblastoma and P27 tell a really similar thing. And what that is going to be, what they're really going to be saying is that when the light turns green, there's positive feedback loops. So the light gets brighter. Like that, right? At first it's a red light, and then you're like, is that green? It's starting to turn, like a couple pixels are green and more. So there's these stories about positive feedback loop. And what that's going to mean is we're going to activate some of the CDK, and it's going to help activate more of the CDKs, right? There are thousands of these CDK molecules in the cell, and we want to activate them all, right? Because we want to get, it's a big job. We got lots of phosphorylation to do, and we got all these guys, and they're inactive, and we want to switch them all on. Okay, so the first, yep.
The retinoblastoma does activate some of the
Yeah,
CDK.
we'll see. It's these first two fit into that, and we'll see them. Now, the book talks about them and has a figure. So the first one is just P27, and P27 is, see on this list we have, there are several of these, and we're just going to tell especially a story about P21 and P27. Right, start is complicated. The small inhibitory proteins, complicated. Here are two simple stories. P27. That's its name, and this is what it does. So a lot of cells at start, the light is red, and it's red partly because P27 is there, and we've said it just is an inhibitor. It's like a school patrol. No matter what else is happening, it's stopped. So what this figure is saying, and it happens over time, and it's a positive feedback loop. And all it's saying is that if you got some CDK active, that active CDK can actually phosphorylate P27 and get it destroyed. And so it's just that thing where the light looks like it's turning a little bit green, and it just turns more and more green, because if we can get a little bit of the CDK active, it helps activate more. So that's the total P27 thing.
What's
Right?
the takeaway? So
Well, the takeaway is you're in your car at the front, and you start seeing the red light turning green, and the school patrol is out. That's P27. And you're like, just get out of the intersection. Get out of here. And all the cars behind you are like, yeah, go, Allie, get him, go. Get him out of there. We got to get to work. So it's a positive feedback loop where school patrol is saying stop, but when a car starts to see, one of the CCKs starts to see a green light, it's like, let's get going. So it's a positive feedback loop. And so, this is a still drawing, but it's showing something that's a dynamic story. And it's very common in science for feedback loops to occur, and it's a little bit hard to represent them. But this is a pretty good one because it's just like, it's off, but if we somehow manage to activate a few, they can, that one of their targets is CDC27, and they get it phosphorylated and destroyed. And that just activates more,
so
like
how
that.
does it get phosphorylized with P27?
Yeah, the CDK.
Okay.
So always remember, CDKs are cyclin-dependent kinases, and we can learn some of their targets. And we just learned, well, MCM is a target. So when CDK gets active, it phosphorylates MCM and the cells start to replicate. At the same time it's doing that, the CDK is phosphorylating P27, if it's there, inhibiting other CDKs. We gotta get the team going, is the attitude. You know, come on, you guys, get out of bed.
Keep doing it.
Shut
Keep
up,
it.
Dave, do not yell, do not yell. Yes, yeah, no, actually, some of the other faculty said, we gotta put like a sort of a thing in his throat so he can't yell. It's like, make his voice bad, and it's like, yeah, but it's a positive feedback loop is we're a team and some of us are sleepy and one of us is, come on, let's go. So it's satisfying to see that. It's just a simple example package, right? And we're trying to do that with START to just put together some simple packages to get a feeling for some of the things that make START complex is their feedback loops, okay? Somebody had another question? Yeah?
So this is kind of giving the impression that CDK is working as a phosphatase.
No, it's adding a phosphate here.
Oh, it's adding.
Yes,
Okay, got it.
yeah. So the CDK is a kinase and it's adding a phosphate.
Got it,
And
yeah.
the phosphate targets the P27 for destruction.
Just for that.
Yeah, like get out of the intersection, you know, like that. Yep. And let's see, Brooklyn or somebody else was maybe
That
had a
was
question.
Pretty
Okay.
good, yeah.
That's it. We're done talking about P27. It's one example of several small inhibitory proteins. There are lots of stories. The book tells some of the other ones. That's a good one because it's just like that. And it could fit in easily where you say, what are the targets of CDK when it gets activated and the kinase starts phosphorylating? And you again would say MCM because that's such a satisfying target because that means S phase, DNA synthesis, and MCM plays a role in helping the DNA polymerase find the chromosome, you know, so, yep. And then this one, sometimes the target includes a positive feedback loop, like destroying P27 to help activate the rest of the CDKs and get them all going, yep. So that's a good one, right? And it's just a taste of START. And in START, there's some positive feedback loops, negative feedback loops. Those always make things complicated, regulatory steps, right? Okay, so that's that one, okay? And you see that. So retinoblastoma does something a little bit different, but has a similarity to it, okay? And that is, we gotta get going, okay? So here's retinoblastoma, and we see this figure, oh, it's DNA, and these are transcription factors bound to the DNA. There's a transcription factor called E2F. And the role of retinoblastoma is to inhibit it. See how it's working here? The retinoblastoma is a transcription factor that does not bind directly to DNA. So we call it a co-regulator, and we call it a co-inhibitor. And it just binds on E2F and inhibits it. Okay? So when the light turns green, CDK catalyzes the phosphorylation of retinoblastoma, and retinoblastoma lets go of E2F. And E2F gets to do its job. And we love this because we say, oh, E2F's a transcription factor. What genes, what are the target genes? What genes are activated by E2F? So let's just look at that for a second. E2F is a group of genes that encode a family of transcription factors. Yay, right? We love it. The 3 of them are activators, 6 are repressors. Ooh, ooh, ooh. Right? A family of genes, some are activators, some are repressors. So it got kind of got complicated. We're going to think of the E2F activators only. And it says all of them are involved in cell cycle regulation and synthesis of DNA in mammals. Okay? So the kind of genes, let's see, I think I have a slide for this. I hope I have that in there. Yes. So we also love this, known E2F targets, right? And we call them target genes or response genes, and there could be a bunch of them, right? One transcription factor is present in thousands of molecules in a cell and can bind the enhancer of a bunch of genes, right? This is very familiar territory for us, where we're just going to tee up and just hit this out of the park. Known ETF targets include genes encoding proteins involved in the initiation of replication. So look at that, MCM proteins. We're gonna make more. There's some, we make more, like that, right? And when they get made, we're gonna phosphorylate them. So we're just cranking this sucker up, right? It's a big job to replicate our big genome, right? So that then nucleotide metabolism, we're gonna synthesize the building blocks. We're gonna need a lot of A's and T's and C's and G's, right? Gotta get this factory going, this S-phase DNA synthesis factory going. And look at that thing, it's just like E2F is bound to those target genes all the time. And retinoblastoma is saying, no, not yet. We do not need a lot of nucleotides, right? Retinoblastoma is like sort of like a pestering parent or something like that. We're like repressing E2F. You know, I know you want to get these made, but we don't need them now yet, right? Enzymatic synthesis of DNA. Freaking DNA polymerase is a protein, and there's a gene for it, and E2F is always sitting on it going, I would like to make some DNA polymerase. And retinoblastoma is just like, no. So here again, we get a taste of the actual just direct cause and effect relationships of the biochemistry and genetics of these green light. When the light turns green, one thing that happens is the CDKs phosphorylate retinoblastoma and let go of it. It lets go of the genes it's repressing, and E2F activates it. And we're making more MCM, we're making enzymes. They're all proteins, we've got to synthesize. We're making enzymes, we're going to synthesize a lot of A's and G's and C's and T's, and we are going to make more DNA polymerase. You know, you're just like, yep, that is S-phase, right? It's the biochemistry of S-phase, like that. And so E2F is huge in that transition. When we say the light is green, there are changes. And this is a big player, right? So look at this. So what is retinoblastoma doing? It's blocking it. It's blocking it until that CDK is active. So we see another kind of positive feedback occurring, kind of a let's get this thing going event happening like that, okay? Next thing is that, just to think about this, is that retinoblastoma is an inhibitor of the S-phase, right? We're blocking synthesis of the stuff we need for S-phase. And so, its normal function is to inhibit the cell cycle, right? Just to keep things off, like that. And because of that, retinoblastoma can sometimes be found as a mutation in cancer, where if it's mutant, it's not doing its job, and E2F is just like, let's go. And the cell has a lot of DNA synthesis interest all the time, because it's got a lot of DNA polymerase and MCMs, you know, and synthesis, got a lot of nucleotides like that. So when retinoblastoma is mutant, it's a weird sort of thing where you're just like, yeah, this cell would really like to be in S-phase right now, right? So it happens in some cancers, and some cancers are in the mood to go through the cell cycle because ETF is active. And they're just like, yeah, we already have a lot of nucleotides. If anybody wants to synthesize DNA, oh, we have DNA polymerase, let's do this. Yep.
They don't necessarily just like build up, they might actually like get used incorrectly.
Yeah, they do build, I'd say they do build up, and it's kind of like if you like stockpile like a bunch of toilet paper, you're probably going to end up using a bunch of toilet paper, right? Because there you go, we have all this, let's just use it. I don't know, maybe not the perfect analogy, maybe it doesn't happen. Just throw it out the window. But that's what the idea is. Retinoblastoma's job is to let's not do all the stockpiling for S-phase until we want the stuff made and the job done, right? And so that's retinoblastoma is doing the job, and it's broken, it's damaged. You're leading into S-phase all the time, and that's what cancer cells do. So it's a problem like that, right? Okay, so because of its role, normal role, retinoblastoma is called a tumor suppressor. Okay, and we've talked a little bit about this, but we have these two kind of ideas in cancer. One is oncogenes like RAS and KIT and MYC and ERB, when they're mutated, they stimulate the cell cycle. And then we have some other proteins. Well, sorry, those normally stimulate the cell cycle, and if they're mutated, we can get uncontrolled stimulation. Tumor suppressors normally inhibit the cell cycle, and when they're mutated, we can accidentally go through the cell cycle. So we have sort of the opposite sides of the same coin. Get a problem with cells that proteins that normally activate the cell cycle, or you can get a problem with proteins that normally inhibit the cell cycle. So we got some tumor suppressors, and one of them is retinoblastoma, like that. And if you want to remember it, like a lot of people call it RB, like that, RB. And here's a story, a person you don't know because I live somewhere else. But when my kids were little, there was another family that had two girls, and the older girl was maybe 10, the younger girl was 7. And they were very cute, and the younger girl, one of her eyes was closed, and it just looked like her eye was sleeping. And she just had one eye, you know, and I'd never ask anybody about any aspect of their body type because that's how I was raised. So I was just like, great, we'd have fun playing with those kids like that, okay? One time, the dad knew I was this kind of scientist, and he said, just off to the side, he said, she's a carrier, she has RB. And he meant it was mutated, that she was born with one of her RB genes mutated, like that. And if the other got mutated, they'd be in trouble. And one place, apparently, and I think it was where it was first found, and that's why it's called retinoblastoma, right? We've seen that these proteins seem to play a kind of more prominent or dangerous role in different tissues, and apparently in the eye, this retinoblastoma gene is apparently especially important for inhibiting the cell cycle. So this little girl had one missing from birth, and if she got a mutation in the other one, cancer would be on the way, especially in her eye. And I think what that meant is that she started getting cancer in that eye, and they just removed it, right? Just that one, because that's where she lost the activity of that one retinoblastoma gene. And we don't know, but she could, at some point, get a mutation in her other eye, right? Like that, just sometime in her lifetime, like that. So it's an example that's, for me, very memorable, and probably for you too, because here's this little kid running around, got one eye closed, right? Because I think they just removed that eye because it started making a tumor. And I think that's why they call it, I know that's why they call it retinoblastoma, because that's where they found it, and it's a tumor suppressor. So you gotta have two good copies, one from mom, dad, just to be good protection. You lost one, I think she lost one, a carrier means, you're born with just one was damaged. Lost the other one, boom, like that, right? So, running in their family. He had an older sister, 2 eyes, 2 parents, 2 eyes, right? So we don't know the genetics of that, but it's a chance thing, right? To pick up that mutation in that other one thing. They're vulnerable, I think. Members of that family are vulnerable, and that's the idea of a carrier. So lots of stuff. This is the entry to lots of stuff in biology, like genetic counseling and talking to that family about what's going on with their health. Yep. Yeah.
I'm confused why it's not considered like a tumor activator, because I thought we were just saying like if RB is mutated that you're going to be making more for S-phase related things. Like why?
Yeah, so the way to think about this is that this thing is going to help cells go into S phase because it's got target genes that are involved in S phase. This thing is playing part of the red light role. It's saying no, no, no.
So it's a tumor suppressor if it's not mutated?
Yes, its normal function is tumor suppression.
Okay.
Yes, and that's, tumor suppressors, most of the time random mutations damage a gene product, right? And so tumor suppressors would be kind of vulnerable because if this got a mutation, it probably would damage its function And its function is to try to suppress the cell cycle. So these are they're famous examples and satisfying package examples to tell this story that adds flavor to START, but also to cancer biology and normal cell cycle regulation. Retinoblastoma, RB. Yep.
It's just like not phosphorylized by something else, so it's an issue somewhere else.
Is it not, is it phosphorylated by something else? I don't know. Yeah, this is our story, just that package right there, boom, like that. So, another target, right? When CDK gets activated at start, it will phosphorylate MCM, help the cell cycle get going. It will phosphorylate P27 and get that destroyed, if that's happening in that cell. And it will phosphorylate RB and remove that suppressor and further kind of do this auto positive feedback loop. Let's ramp up here, folks. We gotta go. We gotta, get stuff organized and start copying chromosomes now. Yep. Okay. Yep.
In this one, they have the CDK4/6 and cyclin D. Is that also referred to as MPF?
No. MPF is for the G to M transition, and that's at the other time in the cell cycle where the proteins involved are actually called CDK1 and cyclin B.
It's only CDK1, right?
Yeah,
Okay.
over there.
Yep.
And we don't need to worry about the letters and numbers, but just recognize that the CDK and cyclins are different at G2 to M than they are at start.
Yeah.
Yep. Yeah, okay. So, there's that. All right, then the last two, let's see, where's our list? And so, that's retinoblastoma. Then, the last two on our list are actually together, P53 and P21. And we'll talk about P21 by talking about P53. So, P53 is a tumor suppressor. That's going to be another, it's going to somehow inhibit the cell cycle. And we're just going to say now, just to jump to the, bottom line, P53 is a transcription factor, and one thing it can do is it can bind to P21 and activate it. And P21 is an inhibitor. So let's just go to that slide and draw a circle around this. Rip. Oh shoot, my little thing turned off. Okay. So there's another thing where we just want to circle part of this figure that's from actually 20, but it shows nicely a really simple story. And the story is that at start, there is a DNA damage checkpoint that we haven't talked about. So it's another thing that makes start more complicated, right? Start normally is just like, hey, should we get going? Yeah, let's get going. When that happens, there's a checkpoint that says, well, let's not start replicating DNA if there's DNA damage. Let's hold on, folks, and get that DNA damage repaired and then go into S phase. So when there's DNA damage, this protein, P53, gets activated. And it's a transcription factor, and for us, it binds to an enhancer of this P21 gene and activates its expression, and P21 is a small inhibitory phosphate protein. Let's see, where do I have that figure shown here? So we're talking about two of those on our list at once. That's good. And P21 is one of those. So P53 does a couple of things, but one of them is it can stimulate expression of this protein P21. So that's why P53 is a tumor suppressor. And it's a tumor suppressor because it will stop the cell cycle by getting that thing made. And that thing, remember, is just the scoop-patrol person. It's like P53 says, get out there, you know.
Okay, so that CK1 is the P53?
Yeah, so, you know, I don't know, we saw in the book, the author uses this little phrase here, I never heard that before. People really call that yellow thing, they really call them small inhibitory proteins. Just because they are small and they are all inhibitory. And we are gonna learn about two. Our author just drops a list of them on you, you know, cause, you know, they're not all, we're not all gonna learn about all of them, but we got two nice stories. P27, we did that one, here's P21. And our key thing for P21 is we could draw in the second exam or the final, draw a drawing of the P21 gene. And it would look like this. You know, and we've done this a lot, right? There's the P21 gene. Here's an enhancer, and this enhancer has P53 bound in the presence of DNA damage. And that's how the cell cycle stops. I'm gonna make a stop here. Stop. Because of DNA damage. So we got DNA damage, activates P53. P53 stimulates the expression of P21, and P21 stops the cell cycle. We have another short pathway that's different. The one we talked before about was at the G2 to M transition. That DNA damage checkpoint was regulated by CDC25. This is a different story. It's over at the other time and place in the cell cycle where we're regulating start. And our major thing about start is just we gotta turn light green. And this is saying, go ahead, turn light green, but if there's DNA damage, we are not going into S phase, folks. And the way we're gonna do that is when there's DNA damage, we'll activate P53 and we'll get it to activate expression of P21, and P21 will just block it. And what happens then? Is CDK active? No. Does MCM get phosphorylated? No. Does P27 get phosphorylated? No. Does retinoblastoma get phosphorylated? No. CDK is inactive and it's just stopped, right? These small inhibitory proteins can just stop and keep the light red because they're like a scoop patrol, and you'd be like, you go ahead, look at the green light. Look at it and honk at me, Ellie, and get out of your car and push me. Scoop patrol is a small inhibitory protein. It's like safety first.
So when P53 is activated, it needs to go through the full gene expression for P21.
Sorry, what?
When P53 is activated, the gene P21 needs to get transcribed.
Yes. Yep.
And then
Yep.
made into the small
Yeah.
inhibitory
So we could show, we can have a drawing on the exam and say, draw the gene P21 in the presence of DNA damage and the absence of DNA damage. And in the presence of DNA damage, you'd say, well, P53 is gonna be bound there, and we're gonna be making RNA, and that's gonna get translated into protein. And if the DNA is not damaged, P53 is not active, so we're not doing that like that. That's the molecular switch, right? P53 is a transcription factor that gets switched on and off, and who switches it on and off is DNA damage. DNA damage is there. We need to activate P53, right? Yes.
The CDK2 still right now, I know you said last class, and I'm just kind of forgetting, like CDK2 is for this part and CDK1 is for what part?
G2 to M transition.
Okay.
And so, let's just go to this, and this might be helpful, and then we'll have a little concept chat action. And that is, where's that figure? Dang it, where'd it go? There it is. Okay? So we're back to this figure, and now we're going to say that this figure shows, it's just showing another way So we learned about P53, and this shows another way of regulating that DNA damage. So I just want to show you that it's just, that's why that figure is complicated. This one is the DNA damage checkpoint at start, and it's showing another pathway that includes CDC25. So this gets confusing, and right, what did we say at the start? Start gets confusing. A lot of stuff going on. This pathway is active at the G to an M transition when there's DNA damage. This pathway is one of the pathways that's active at start. However, this pathway, the book is showing, and it's confusing, so I need to fully explain that figure. I won't ask you about that pathway, I'll ask you about P53, because it's a different story, an additional DNA damage response, right? Okay? So that's confusing, but again, it starts confusing, and we're wading through it, and don't worry about this one, except to appreciate that we don't have to learn about that pathway. We learned about that pathway, and it was at G to M. And when we go over to the DNA damage checkpoint at start, we talk about P53, like we just did. P53 is super famous. There's a list that was made several years ago of like one of the most studied proteins based on number of scientific publications. What is the top 10 most studied proteins in the last 20 years, right? And you know, insulin, you know, hemoglobin, you know, name proteins you know, none of them are on the list. P53 is. It's just crazy how focused people are on P53. That's what it does. It does some additional things, but it's a transcription factor, and one of its targets is just make P21. It's got DNA damage, you should make P21. It will stop this thing now, right? Like that. People call P53 the guardian of the genome for that reason. It's ridiculous how people have T-shirts that say guardian of the genome, and it's P53, and you're like, you know, you go to a concert, wear my shirt, right? That's the one we really want to understand, because it's simple. And there was a question that somebody had or something.
Yeah.
Yep.
So does P53 work on single-stranded breaks only, or is it double-stranded as well?
I don't know. Yeah, there's different. The book is trying to give some flavor of how these might be activated a little bit differently depending on different kinds of DNA damage. And I don't know that much about it, but my understanding is that it's kind of you know, there's specific types of DNA damage, and that doesn't just break down clean, like you'd be like, is it just limited to that? You know, because there are multiple pathways to get P53 activated and so forth like that. So I just kind of gloss over it and just say DNA damage, just because if you get into the weeds really fast. Okay, let's see if we have time. Yeah, we're just gonna have a goodbye concept chat like that. So get together with a partner and talk about 3 things, because we talked about some things that are important today in class. And I did hand out the sign-up sheet, so that's out there. And so just find somebody and just be like, oh man, we're gonna have to get so many tattoos. Right? What are 3 tattoos that we could get just from today? Hi, how's it going, Riley? Wait.
Which is interesting.
I only just really proud of that one.
I think it's cool that the original left way was kind of represented.
Let's go back.
One that happens.
So this is a long and all
Cool, all right. All right, so that was Riley. Riley just got here late and didn't want to interrupt. Darn it. So he's going to be here on Tuesday. What day is it today? Thursday. Yeah, he's going to be here on Tuesday.
You're asking me a question I don't know?
Let us talk to Hannah. No, we've heard from Hannah. Amal, what is one thing you guys talked about you thought was important? Oh, I've been to a lot of concerts. Have I been to a lot? Sorry, what?
Start regulation, we're talking about like the federal Western one.
Yeah. So START is our big thing, right? We've really been focusing a lot in our course, and those pathways that lead to activating the cell cycle. And it's complicated, but we have to try to divide it up into some pieces to build a good foundation, right? So I'm trying to do that, and we're back at START again. We also talked about the G2M transition, right? That's a small package for us. And then back is just, we're almost done talking about START, but just some more pieces, and some of them are just super famous. Like if you said, oh, you taught a course in cell molecular biology and you did not talk about B53? How'd that happen, David? Because that happens, because down at Maine Health Institute for Research, those scientists ask students like Eva, who's down there, and others who are down there, and they're like, hey, what'd you learn about P53? And you're like, I never heard of it. And they're like, David, what are you thinking? Okay, what's another person who is, Jeremy, what's one thing that we
We
talked about?
talked a lot about the retinoblastomas and their activation of the E2F, which kind of leads to this cascade of other gene expression.
Yeah, man, E2F just is a transcription vector, and it has just such satisfying target genes, right? We gotta make some more MCM.
And
Gotta make
so-
some DNA polymerase, you guys. Come on.
Yeah, so retinoblastoma is kind of easy to remember because it blasts a whole bunch of genes out. So
Oh, little mnemonic there. Yes. Yeah. The original term being though, I think tumor,
Yeah.
blast the tumor out too. So yikes. Yeah. Okay. And then Eva, what's one other thing you guys talked about?
Just we focused also on 27 and the positive feedback loop, the active sequencing of the complex. When it's activated, it further activates itself.
Yeah.
By release.
Yeah.
And
Further activates more of it. So there's a bunch of CDK in the cell, and some of it gets activated, and then it helps activate more of it. Yep. Okay, we gotta be done. I bet this might be a class meeting that you watch again this week. You might just watch it again and be like, oh yeah. There are packages, right? We're putting together little packages, little examples to understand. And each example has a little pathway, some small number of steps. And so you can make a flashcard about the guardian of the genome, P53.